Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello students, welcome to the class. Now, in this class, we shall start discussing in detail with the phases of prophase 1 in meiosis 1. I already told in the last class that apart from all other phases, the prophase 1 plays a significant role in the meiotic cell division because very important events like crossing over, recombination, pairing of homologous chromosomes are taking place in prophase 1. So now we shall start discussing with the prophase 1 in detail. The prophase 1 is subdivided into 5 subphases as leptotic, zygotic, patatic, diplotic, and diakinesis. These are the 5 subphases of prophase 1. And to start with the leptotic first, before entering into prophase, in mitosis also we have discussed that the cell has undergone the preparatory phase, that is the interphase. So I will draw a cell which is in interphase now. So here is the cell which is in interphase. In interphase, that means it is just completed the interphase. The nuclear membrane will be present and there are replicated centrioles present here, and the chromosomes are not formed, that is the chromosomes are present in a complex network. We call it as the chromatic network, that is the cell is in interphase now. When the cell enters into the first phase of prophase 1, that is prophase 1, when it enters, the very beginning will be the leptotic phase. The leptotic phase is otherwise called leptonema. Both are the same, or it's called leptonema. When the cell enters into the leptotic phase, here the nuclear membrane will be present as such. There is no change in the nuclear membrane. So here the nuclear membrane is present as such, and there are centrioles which have started moving to the opposite pole. Not exactly they moved apart, they just started moving to the opposite pole, and there is a formation of the spindle asters, and also between the centrioles there is a formation of spindle fibers begin. Spindle fiber formation has just begun. Remember, they are not, the centrioles have not moved apart here. They just started moving to the opposite pole. Just traveling, they started, and there is a formation of spindle fiber between the two centrioles. And since the nuclear membrane is present as such, this is nuclear membrane, as such it is present, the other organelles associated with the nuclear membrane like Golgi apparatus, endoplasmic reticulum, they are also present. They have not disappeared in this stage, that is in leptotic. And in leptotic, there is a condensation of chromatin. There is a condensation of chromatin network happens. As a result, there is a formation of chromosomes. There is a formation of chromosomes, and the chromosomes which are formed in the leptotem are very thin. The chromosomes will be very thin because the complete condensation of chromosomes has not happened here. There is just the the chromatic network has undergone the condensation, and the chromatic network will be condensed in a loose pattern. As a result, the chromosomes appear very thin, and that is, the appearance will be like this. So here is the chromosome. This is one chromosome, and this is another. This is another, and this is another. The 4 chromosomes I have taken, and whole meiotic division I will be having a track of 4 chromosomes. What happened to them? And on these thin chromatin fibers or the chromosomes, there is a formation of nodule-like structures. Nodule-like structures are formed on the chromosomes, and these nodule-like structures are called as chromomeres. They are called as chromomeres. I will repeat now. In the leptotic phase, the nuclear membrane is not disappeared, and the organelles associated with the nuclear membrane are also not disappeared. The centrioles have just started moving towards the opposite pole, and there is a formation of spindle fibers between them, and because of the condensation of chromatin network, there is a formation of thin chromosomes. Within those thin chromosomes, the chromatins are loosely arranged, and also at some regions on the chromosome, there is a formation of nodule-like structures. Those nodule-like structures are also made up of chromatin network, no doubt. And those nodule-like structures are called as chromomeres. And these chromomeres are very important during the association or the pairing of homologous chromosomes. And if you observe here, I have written the chromosomes in attachment with the nuclear membrane. I have not written it suspended within the nucleus or the nucleoplasm. I written it in attachment with the nuclear membrane. There is a reason for that. In this stage, the chromosomes are polarized. The chromosomes are polarized, which means they are attracted towards the nuclear membrane. The chromosomes in the leptotic phase, they are attracted towards the nuclear membrane, and this stage is called as backward stage. This is called backward formation. That is the attachment of the thin chromatin fibers or the chromosomes in leptotem to the nuclear membrane is called backward formation or the backward stage. So these are the events which are taking place in the leptotem, and for convenience I will write the 2 chromosomes in similar color. So I am writing 1 chromosome in yellow color, 2 chromosomes in yellow color, and 2 chromosomes in pink color, which means the color represents they are the pairing homologous set. That is, this pink and this pink are one similar kind, and these 2 are similar kind. That represents, because it will be easier for you to understand the pairing of homologous chromosomes in the next phase. So these are the events in the leptotem, and the next stage is zygote. Zygote, or it is called zygonema. zygote or zygonema. Here also the nuclear membrane will be present as such. The nuclear membrane will be present as such, and the centrioles, they are traveling, and the asters also present. And here in the zygote phase, there is a pairing of homologous chromosomes takes place. This is one chromosome and this is another chromosome, that is of the homologous set. In the last class I told what is meant by homologous. Homologous means they are having the same kind of genes, but one is from the maternal, another from the paternal. That is, one chromosome in a homologous set we received it from father and another we have received it from mother. So that is called homologous set. And remember, I told in the last class also that these two chromosomes will not stay together always. Always they not stay together. Here also I have written it very different. That is, apart they are present. That is, in pair they are not present. The pairing will happen only in the zygote. That is the key event of zygote. What is the key event in zygote? The pairing of homologous chromosomes. The pairing of homologous chromosomes will take place, and the process of pairing of homologous chromosomes is called the process of pairing of homologous chromosomes is called synapsis. Synapsis, or it is called synthesis. It is called synthesis. Synapsis or synthesis, both are the same. And after pairing of homologous chromosome, in a pair there is presence of two chromosomes. So there is two chromosomes present. This is one chromosome and this is another chromosome in a pair. So there is presence of two chromosomes, each having two chromatids. Actually I have written here one, but it is not the case. Already the DNA is got replicated, so there will be formation of two chromatids in future. So here each of the chromosomes are having two chromatids, so it is called dyad. Chromosome dyad. And in a homologous pair, there is presence of four chromatids. Each chromosome is having two chromatid, means in a homologous pair there is presence of four chromatids. Hence this is called as tetrad. Chromosomal tetrad. Tetra refers to four. Four here is four chromatids. In a homologous pair there is presence of four chromatids. So we refer the term tetrad. And we could also refer one more term here, bivalent. Bivalent here also refers to two chromatids in each chromosome, or two chromosomes in a pair. So we will refer different terms: chromosomal tetrad, bivalent, or the dyad. And here in the homologous pair, that is, in a homologous pair of chromosomes, there is presence of two chromosomes, and I already told one of the chromosome is paternal and the other chromosome is maternal, and they will come together for a purpose. They simply just not come together and stay there. They stay there or they come together for a purpose. There is a purpose for pairing of homologous chromosomes, because the purpose is to carry out the process called crossing over. The process of crossing over is because of the pairing of homologous chromosomes. So to cross over, that means the part of this chromosome got exchanged with the part of this chromosome. For exchanging that matter, the distance between the two chromosomes must be too less. The distance between the homologous chromosomes is too less. Negligible distance is present, and that distance is measured to be approximately 100 nanometer, which is a negligible distance. So if that distance is present, then only it is possible to exchange the part of chromosomes. And here the homologous chromosomes are present in the nucleoplasm. That means it is a liquid. So there is a chance of separation of these chromosomes, right? So if we suspend something in a liquid, they got separated, they will not stay together. But even the two homologous chromosomes are present together in the nucleoplasm because of the presence of a synaptonymal complex. So to define it, the synaptonymal complex. Synaptonymal complex is a ladder-like proteinaceous structure formed between, I'll write it here, formed between the homologous chromosomes. That means there is presence or there is an arrangement of different types of proteins between the two homologous chromosomes, and the arrangement of those proteins is very peculiar, and it will help the two homologous chromosomes to stay together till they got the process called crossing over or the recombination. Till then, there is a formation of stable proteinaceous complex which is called Synaptonimal Complex. It is formed between the two homologous chromosomes. And remember, and here there is no formation of the bivalent, that is, two chromatids have not formed. The chromosomes will be having one chromatid. There is a thin line. That's it. So now, to study this synaptonymal complex, so that is, if we from here to here there is presence of synaptonymal complex. So entire thing will be having synaptonymal complex. So the structure will be same. So to explain that, I am taking only this part. If we take this part and if we observe under microscope, here is the chromosome, one homologous chromosome, and here is the another homologous chromosome. What we see here, it is only this part I have written, so I will just write it in the same color so that it will be easier for you. So here is the one homologous chromosome, there is the another homologous chromosome. Between these two, the space is synaptonymal complex, where there is arrangement of different types of proteins. And here, in attachment with the inner part of the homologous chromosome, the inner parts of the homologous chromosomes are having the lateral elements. These are called proteins. They are made up of the lateral elements. These are called lateral elements. Lateral elements, both of them. And at the center, there is the same kind of structure present, which is also made up of the proteins, and it is called central element. Central element. And since there is presence of three elements, or three types of three proteinaceous structures are present, this is called tripartite. Tripartite refers to two lateral elements and one central elements. That is, they are not different structures, they are the proteins. They are arranged, the lateral elements are arranged in attachment with the two homologous chromosomes, and at the center there is a central elements which are arranged. So it is to give the stability. And all of these, that is the space between the lateral element and central elements, is filled with the transverse proteins. The space between the lateral and central elements is filled with the transverse proteins. So this is the transverse proteins, or we can say it as transverse fiber. This is called transverse fiber. This is called transverse fiber. So this is how the two homologous chromosomes stay together. This structure is responsible for the stable attachment or stable association of two homologous chromosomes, and this is the structure of Synaptonymal Complex, and it is called tripartite because of the presence of two lateral elements and one central element, and these are connected with each other with the help of a transverse fibers, which are also proteins. These are the longitudinal proteins, and these are called the transverse proteins because of the arrangement. So this is how the two homologous chromosomes will stay together in the zygote. That is the key event, that is the synapsis. The two homologous chromosomes will associate in the zygote, and there is a formation of Synaptonimal Complex for facilitating the close association of two homologous chromosomes, and we will refer different terms for this homologous chromosomes like dyad, tetrad. So these are the things which are happening in the two phases of the prophase 1, that is leptotic and the zygote. We shall continue with the other phases from the next class.